Hello again. I'm Maddie.
I'm Steve.
And today we're going to take you on the tour of the Zebrafish lab. Okay, a couple of interesting things about zebrafish. We can genetically modify them to more readily study how cancers and different types of treatments behave in them. And they have 70% of the same genes as us. So that's lower than mice, who have around 85% of the same genes as humans. But of course, the zebrafish are a fraction of the cost and grow way faster. So they even have clear ones, so you can see what's going on with their vital organs at any given time.
Because you can buy them at the pet shop for $2.50, but they're very similar to us in terms of their genes. And we now know that in cancer, cancer is really a genetic disease. That doesn't mean it's always an inherited disease, in most cases it's not, but it's a genetic disease that in those cancer cells, some of those genes, something has happened where you have mutations or rearrangements of your chromosomes, or you're missing a gene, or something has happened that make those cells divide in an uncontrollable way. So if we have a model system like zebrafish that have similar genes, then that allows us to be able to look at normal developmental processes and how they go awry and get sort of screwed up and cause diseases like cancer. And so the fish has the added advantage of it being these larvae being transparent, so that we can look and see what's happening inside.
Okay, now that you have a little bit of a base of knowledge, we're going to go to the secret lab. Kronk, pull the lever. Wrong lever!
No. Try this one. No. Try this one. To the Zebrafish lab!
Pull the lever, Kronk. Wrong lever! Huh? Why do we even have that lever? Get out of my way!
Please remain seated and keep your arms and legs in at all times.
So when you go into the lab, you have to get your bottom of your feet sprayed so that you don't contaminate the lab. Dax, are you interested in learning about cancer research? What about Goose? No, he's not interested. Okay, let's go to the lab.
Nutrition for the first few days of life. It's sort of like the placenta for the zebrafish. We have a number of different mutants, zebrafish, that we use. So these mutants are called Casper mutants. They're fish that have no stripes. They're completely transparent. People say you can read the newspaper through these fish. You can see, and you'll see them in there, their hearts are pumping, and you can see the females, the eggs, and you can see food moving through their GI tract. And we use these for our transplant studies because they're completely transparent. Then we have a number of genetically modified lines that, for example, express certain fluorescent markers. So these fish express this green fluorescence in all their blood vessels. So you can see in the embryos here how the blood vessels are outlined in green. And you can imagine that if we put human tumor cells here that were red or blue, we could follow and see, do they spread through the blood vessels? Do they develop more blood vessels? Where do they go? That kind of thing. And we can do this pretty easily.
What it is, it's a combination of two homozygous mutants. One is called Nacre, which lacks melanophores in the skin, and the other is Roy, named after Roy Orbison because of the dark sunglasses, so they have these black eyes, which lacks iridophores, and you combine the two, and they have this see-through appearance, and it allows...
So here's an example of what researchers can do in real time for a patient. And there's a hospital right next door to the lab. In fact, the doctor you saw at the start of the video is a pediatric oncologist who splits his time between seeing patients who come from all over Atlantic Canada to see him, between treating patients and leading cancer research. So they can take a sample or biopsy of cancer from a patient, implant it into 10 different zebrafish at the lab next door, and test multiple courses of treatment on them. So they'll color the cancer a bright color. Say they try 10 different drug combinations and see that drugs 1, 3, and 6 are causing a reduction in the number of cancer cells, but the one that is most effectively reducing the number of cancer cells also has an adverse effect on the lungs. So they can take that into consideration, think about what other combinations or supports could be provided if that drug is selected as the course of treatment. And so they can do a whole second round of testing within a few days because the zebrafish's life cycle is so fast. So with mice, for example, this process would take much longer, and being able to see the effects of the different drugs is also a lot trickier than in the case of zebrafish.
The time-lapse experiment overnight for 12 hours, and what we can see is we can see red Ewing sarcoma cells actually moving and migrating into the vessels, and that's one way that we can kind of track metastasis and aggressiveness and see if the cancer cells will actually home or migrate to a specific tissue within the zebrafish. So if I can bring this back to the beginning, what's really cool about this video is you'll actually see one cell come out and travel down, but then it will go back and actually recruit about 4 or 5 other cells, and then you'll see them drop down as a clump.
That's amazing.
Which is really cool. So there's some sort of cell signaling going on there, and it's cool because we can actually visualize this.
Was that the same cancer? Was that
it's a
the
Ewing
sarcoma?
Ewing sarcoma, yeah. I can just bring it. Oh, it restarted again. So we can actually watch individual cells move and how they can interact and kind of recruit other cells.
Wow.